The eLife podcast from eLife, the open access journal for outstanding research in the life and biomedical sciences. Online at elife.elifesciences.org.
Hello, welcome to episode 13 of eLife's podcast, produced by The Naked Scientists. I'm Chris Smith. This month, exercise makes blind mice see, why hybrids don't make their parent species become extinct, and a way to make the entire population of mosquitoes resistant to malaria. First though, a team of scientists have successfully harnessed the power of computer face recognition technology to turn it into a diagnostic tool so that individuals with very rare syndromes that are usually associated with characteristic facial features can receive accurate diagnoses. Christopher Nelliker is at the University of Oxford.
Some rare diseases are so rare that a single clinician might not ever see them in their entire career. And the reliance has been on identifying multiple people with the same disease when there might be less than 10 people across the entire world alive at any one point. So we tried to create a computer vision approach to teach a computer to analyze faces in photographs to try and find and cluster these patients by which disease they might have.
Was this literally going up to people who have diagnoses, taking their photo and then training the computer, this is what condition X looks like? Or can you be cleverer than that and go to online databases where there might be many images of these people and build your tool that way?
So we went online and collected publicly available photographs of people with well-established and relatively common rare diseases that we could then collect lots of examples for. Because with machine learning and computer vision, you're reliant on many examples to get accuracy. So we got around about 100 to 200 images for 8 different syndromes, and we used those to train the computer what to look for.
And these are all disorders that are manifest with changes to the way a person's face makes them look?
So not all rare diseases will have changes in their facial structure, but in the development of the head and face, a large fraction of our genes in our genome are used. So it's quite likely that a mutation might disrupt some part of development of the facial structure as well. So it's been estimated that maybe 40 to 50% of rare diseases at least have some sort of manifestation in face or cranial structure.
And so what you would present your system with a snapshot. It would draw out these features by analysing the face, and then it would compare its snapshot readings with your database of many different faces from individuals with known diagnoses and see which features it shared most in common with those faces, with those diagnoses, and then return you this is likely to be condition X.
Yes, so it could be used in that way, but the current approach has just been a proof of principle, so it's not sufficiently accurate at the moment to be considered a diagnostic tool. We consider it more as a way of narrowing the search space for possible diagnoses, but it does this by comparing between people it's seen before.
What about the fact that you're just using two-dimensional photographs? Could more accuracy be delivered by looking at three-dimensional images if you were to take stereoscopic pictures of an individual and analyse that?
Absolutely, and so there has been a lot of work into analysing rare syndromes with 3D imaging previously, and they get very detailed data. But our approach tries to expand into more accessible data because everyone has access to a camera or a scanner or a digital camera. And so it is easier and more accessible in future to try and use normal 2D photographs. And so we don't have as higher accuracy as for 3D imaging, but with the machine learning we've been using, we've been approaching the discriminatory or classification accuracies of 3D imaging for the 8 syndromes we've been using.
So how would you see this being deployed in future? Would this be a tool that, say, a hospital doctor could take a snapshot of a newborn baby and feed it into your system, which would remotely analyse it and then return a possible diagnosis or list of diagnoses for individuals that clearly do have a problem, but the doctor may not know what they are?
Yes, so that's precisely how we see this being deployed in future through normal healthcare services as an expert tool for clinicians to narrow diagnosis when a rare disease is already suspected.
Christopher Nelliker. If an animal has an eye kept closed from birth, after a few weeks, even if the eye is subsequently opened again, the brain nevertheless remains blind to input from that eye forever. The same happens in humans who are born with eye problems. If these aren't corrected quickly, the individual is destined never to see normally with that eye. But now Michael Stryker has found a way to dramatically turn things around.
A few years ago, we discovered that when mice start to run, their cortex goes into a high gain state, and in which the neurons respond much better. And the question we wanted to ask in this paper was whether this enhanced response would allow more neural plasticity, that is, would allow the animals to change the way their brain responds much faster in response to a certain kind of activity. Would they learn better? Would they recover function in their visual system if their visual system had developed abnormally?
When you say they're in a high gain state, do you mean that literally if you record from groups of nerve cells firing in the cortex, you see more firing, or do you see bigger pulses, bigger amplitude?
We see more firing. More of the neurons fire much more strongly, about twice as much when the cortex is in this state. And we think this state might be important for things like attention, but at least in the mouse, their visual cortex goes into this state when the mouse starts to move through the world.
Part of me is wondering if this is the reason why some people, I mean, it sounds a bit lame, but some people say they like revising when they go for a jog, and they'll listen to some learning tape and they'll say they remember more if they go jogging. Do you think the same applies to humans?
I don't know whether the same applies to humans, and it's something we're tremendously interested in. We're beginning to study now. Several of my colleagues tell me that they like to read papers on the treadmill because they remember them much better and understand them much more quickly.
I'm calling to mind the memories of medical school lecturers pacing up and down at the front of their lecture. You never see a lecturer stand still unless they're highly polished, or a comedian stay in one place on a stage. They always walk around, and one wonders whether this is playing out in the same way as your mice do.
I suspect it may be, because I feel the same thing when I lecture. I would much rather walk back and forth and point at various things and be physically active rather than to stand there like a statue.
So what did you do with the mice? Because obviously you can't get mice to give a lecture.
What we did with the mice was to treat them like children who were born with a congenital cataract in one eye that obscured the vision in one eye. And so we took mice and closed one of their eyes for a period of several months. And humans who grew up with a problem like this, later, when the optical problem is corrected completely, later these people have lost the ability to see in that eye and are dramatically impaired when they try to look at the world through that eye. They're functionally and legally blind.
So is this because something in their brain has changed, which means their brain is essentially ignoring the visual input from that eye, which it previously would have paid attention to?
Yes, most of the connections from that eye to the visual cortex are lost during a critical period in early life under these conditions. And so the reason the people find it difficult to see is that their eye is not connecting properly to the brain anymore. And the goal of our experiments in mice was to see whether these enhanced neuronal responses we see in the visual cortex during locomotion would allow them to recover much more vision and much stronger visual responses to the deprived eye.
And is that what you find? So you get the mice doing more activity and compare them with mice that do less activity. Do you see that they get more functional recovery in the previously closed eye?
They get dramatically more functional recovery, so that mice that get no special stimulation and stay in their home cage never recover more than halfway to normal. Whereas the mice that are allowed to run while they're getting high contrast visual stimulation, their vision recovers to near normal levels quite quickly within a week.
There's a central sort of tenet in neuroscience, isn't there, which Hebb put forward, which is that things that fire together wire together. So do you think that you've got a very simple situation where you've just got enhanced firing because of the locomotion, the exercise? You present that with a very powerful visual stimulus. This is going to drive cells very hard as well. So you've got the same cell firing with the locomotion and with the visual stimulus, and the connections there are going to reinforce.
That's one of the possibilities, and it's the one that I think is perhaps most likely. But another possibility is that when you have this exercise, you have more acetylcholine, which is a chemical that affects neural transmission in your cortex. You have more adrenaline. So it's possible that some of these chemicals that are associated with this are the signals that mediate the greater plasticity. But the idea I favor is exactly the one you articulated.
And obviously one very important question from a sort of neurorehabilitation perspective. If I took someone who had had a disabling stroke, robbing them of one particular brain function, would the right thing to do be to repeat the experiment you've done with the mice, but with an appropriate stimulus relevant to their injury, and see if you can get, therefore, enhanced recovery for them?
Well, that is the long-term promise of this. And at present, we just don't know whether that would help or not. We intend to study that, but at the present time, we really can't say whether that would be true or not.
Michael Stryker, he's based at UCSF. You're listening to the eLife podcast with me, Chris Smith. Still to come, a genetic strategy to render the entire mosquito population resistant to malaria. Before that, though, the genetics of hybrids. When two closely related species mate and form so-called hybrids, over time, why doesn't this lead to the disappearance of the two parent species? Princeton's Molly Schumer has been comparing the genetic profiles of two closely related fish, and what she's finding is that some combinations of genes contributed by the two parent species don't work well together, owing to the presence of some species-specific adaptations. And although individually their contributions are quite small, at the level of an entire genome, they exert a powerful selective force.
In the past 10 years, we've begun to realize that mating between different species, or hybridization, is really common. And that raises a little bit of a problem in understanding how species or different groups of organisms stay distinct in the face of this genetic flow between them. We study two species of freshwater fish, Xiphophorus birchmani and malinche, that hybridize quite often. But we don't see a lot of evidence that these species are really breaking down. And so we wanted to understand what genetic barriers might be existing between them.
Is this because, Molly, when you get a hybrid, that the hybrid itself then cannot continue to interbreed with members of the individual species, and that stops the further evolution of that hybrid?
So in a lot of species, hybrids are inviable or infertile, but that isn't really the case in these species, which is part of the reason they're very interesting.
So perhaps the hybrids are more vulnerable to being caught by a certain predator, and this keeps their numbers down.
Or they're less effective at foraging or finding mates or any of these sort of things that are important in the wild but not apparent to us in the lab.
So how did you explore this genetically to try to find out what was going on?
Basically, the idea behind what might be breaking down in hybrids is that you have these two genomes in the parental species that are evolving independently, and they accumulate mutations. And then when you reintroduce these genomes to each other in hybrids, they can interact poorly. And to give sort of a specific example of what that would look like, if you have two proteins that work together in the same pathway. And in the two species, they've accumulated differences and they can't work together anymore. Any hybrid that has the combination of those two proteins will be selected out of the population. So then when you go and sample hybrids from the population, you'll see an absence of that combination, which is sort of the footprint of selection on that population. And so that's basically what we did, is we collected hundreds of hybrids from two populations and looked for that signature of missing genotypes.
And what did you find? Were there areas of the DNA which clearly, if you mix them together in a hybrid, were a toxic combination? They just didn't crop up when you went looking for them?
Yeah, so surprisingly, we found 207 pairs of regions that were significantly missing in both hybrid populations. And this was a lot more than we expected. These two species have some clear differences in terms of their ecology. For example, Malinche is adapted to very cold environments and Birchmani is adapted to warm environments. So we expected that we might see a few regions associated with ecological differences, but we did not expect to find so many.
And what does this tell us more generally about how hybrids are or aren't maintained in populations?
So I think the bottom line here is that when you look at hybrids in the lab, you're missing a lot of the selection that's going on. And one of the things that I didn't mention earlier is that even though we find so many pairs, when we estimate selection on these interactions, the selection is actually quite weak. So around, a 2% or 3% disadvantage for that hybrid genotype. And what that means is that in this particular case, many selection effects that are weak are contributing to reproductive isolation between the two species, as opposed to the more classic cases where you have strong selection effects on just a few sites.
Do you think that, because you've said there are lots of little contributions, and so a big number times a small number still makes a fairly big number, which is why you see this effect. Do you think that the same sorts of genes are going to crop up in different species, though? Or do you think that the kinds of genes that exert this fairly toxic effect, they're going to end up being species-specific?
We actually did not see a strong pattern in terms of the kinds of genes that were involved. So there are sorts of different analyses that you can do to see if there's overrepresentation of certain kinds of genes. And we did not actually see that any particular category of genes was overrepresented, which suggests this is kind of a broad effect, that many genes are involved in contributing to isolation in hybrids. And I think that that'll be the case in other species as well. Though in some species, they have found that genes on the X chromosome, for example, are significantly more important in contributing to reproductive isolation, which is not something that we found.
Molly Schumer. Rett syndrome is a neurodevelopmental condition linked to a gene called MECP2, which controls the activity of other genes by binding to DNA and then suppressing their expression. But which of these two processes are key to causing the disease and a related syndrome caused by having additional copies of the MECP2 gene? Laura Heckman has cleverly solved the puzzle by making mice that carry a working, healthy copy of the MECP2 gene alongside an altered copy that lacks either the DNA binding activity or the gene repressing activity.
So in our lab, we study a gene called MECP2, and it turns out that it's really important because we see that if you have too little of the gene, in humans you see Rett syndrome, which is a progressive neurological phenotype. Girls typically have this disorder, and they'll develop normally for the first 6 to 18 months of life, and then you'll actually see developmental stagnation and then regression. So they'll lose purposeful hand movements, they'll develop anxiety and other autistic features, and we'll see late motor deterioration. And on the other hand, if you have too much MECP2, then this presents in a different disorder called MECP2 duplication syndrome. What's interesting about this is that it's also a progressive neurological disorder, and we see some overlap with the phenotype, such as autistic features, but it's also distinct in that we see spasticity and seizures and infantile hypotonia, which isn't seen in Rett syndrome.
What exactly does this gene do?
So we know a bit about what MECP2 does. Its level increases highly throughout development, And so far, what's really known is that there's two main domains. One binds DNA, and then the other represses other genes. And so I think that these work together to regulate other genes that are expressed during development and then throughout adulthood.
So, given that important role, its ability to influence many other genes, it's not surprising that it probably has an impact on the way that the brain develops and functions. But then the question is, well, why does too much of it have this effect, and why does too little of it have an effect? How can you disentangle the two?
Exactly. And that's one of the questions that we aim to address. So to try to figure out what MECP2 is doing in both Rett syndrome and MECP2 duplication syndrome, we chose to study 2 point mutations in the gene, one which affects the domain which binds DNA, and one which affects the domain responsible for repressing genes. And so when we look at these, when it's the only copy of MECP2, we can get an idea of how it affects the function of MECP2. And on the other hand, if we express it with an extra copy of MECP2, then we can get an idea of which domains are required in order to see these toxic phenotypes.
So you can dissect apart which of the two ends of the molecule is involved in each of the two phenotypes, can't you?
Exactly, yes.
And go on then, spill the beans. When you do this, what do you see?
So when we do this, we see that we actually need both domains completely intact in order to see the duplication phenotypes. And this is really interesting since a lot of previous hypotheses thought that maybe just the one domain was responsible and the other didn't really play a large role.
So you need both ends of the molecule to be active in order to do the job of causing the syndrome if you have the duplication problem. But what about if you've got the deletion or the initial mutation just in one copy? What about that phenotype?
When it's in the Rett syndrome context, we also find that completely messing up one of the domains will lead to a null phenotype, and it's very severe, actually. So that means that whatever additional function the repression is handling, it's not enough to overcome the lack of binding to DNA. And on the other hand, when we looked at the repression domain, we actually found a couple interesting things. We saw a milder phenotype, so they still have many of the same phenotypes that we're used to seeing, but not as strongly. And we also found an additional function for this end of the protein. So it was initially thought that it just interacted with other proteins in order to repress transcription, but it turns out that it can also bind DNA.
And what do you think the implication of that is? I mean, what does that actually translate into in terms of our understanding of this process?
So I think there are a few important takeaways from this. One is that we now see that in order for MECP2 duplication syndrome to occur, that you need the entire protein to be functional. The second is that we see these differences in different mutations that have various effects on the individuals. So for example, the previous models, many of them die very early on, and so it's hard to study and hard to figure out how to help the mice and therefore the patients. So these new mice are better so that we can have longer outcomes for potential therapies. Additionally, it's also interesting that putting in a normal copy of MECP2 can rescue many of the Rett syndrome phenotypes. And so this can also become a viable potential therapy in the future.
Laura Heckman from Baylor College of Medicine. And finally, a strategy to deliver genes to entire populations. This process, called a gene drive, could be useful for propagating desirable characteristics into a species, like malaria resistance in mosquitoes, insecticide sensitivity in plant bugs, or even herbicide sensitivity being engineered back into resistant weeds or crops. Harvard's Kevin Esfelt explains how it works.
One particular animal that we would love to alter in the wild is the mosquito, so they can't spread malaria, which is a terrible disease that kills 650,000 people a year, most of them children under 5. We know about particular genes that we can put into mosquitoes that render them unable to transmit malaria effectively. The question is, how can we spread those genes through wild populations? And we started by looking at some results from the laboratory of Austin Burt at Imperial College London, who ran some simulations suggesting that you might be able to spread genes through wild populations if the gene can benefit itself. Now, there are some genes in nature that can do this by biasing the odds that they will be inherited by offspring.
How do they do that?
This particular class does it by cutting the competing version of the gene such that the cell will then copy over the cheater. And Austin Burt suggested that we might be able to adapt these genes to spread traits that we want to through the wild organisms.
So if we look at your malaria example then, if you had genes which are capable of suppressing malaria in a mosquito, and you confer this technology on that gene, then it's going to copy itself within the individual affected mosquito and then propagate through the population because it strongly selects itself.
That's exactly right. Because it is inherited almost twice as often as a normal gene, in fact, that single gene advantage is strong enough that it can even spread if it reduces the ability of the organism to survive and reproduce. The problem was that we didn't have the molecular scissors necessary to make the cuts. And we needed really good molecular scissors that could not only cut whatever sequence we wanted, but could also cut multiple sequences right next to each other to reduce the odds that evolution could come up with mutations that would block the ability of the scissors to cut.
So do you mean, in the same way that when we have an infection and we use antimicrobial drugs, bacteria, other microbes can mutate to surmount the effect of the drug, but if we give multiple drugs at once, because we're hitting different independent targets, it's much harder to mutate each of those targets independently and simultaneously to make an organism that's resistant. You could do the same thing with a malaria mosquito with this technology.
That's exactly right. If we can target multiple sites within the original gene that we want to replace, then we can make it that much harder for it to evolve resistance to our ability to copy over it.
Now, just looking at the nuts and bolts of this a second then. So what this would take then is you have to have a set of these molecular scissors which are going to cut genes. You've got to plumb that into the genome in the correct genetic place or locus which you want it to target, so it then copies itself with the ability to delete any opposing gene and then copy itself in there. But you're going to have to do that not just in one, but multiple places in the genome. Can we do that?
We can. Over the last couple of years, we've been developing a technology called CRISPR that is essentially molecular scissors that can target almost any sequence in the genome, even multiple sequences in the genome. And we've used it to date to edit the genomes of a wide variety of organisms, because it seems to work in just about everything. And the way we do it is we introduce the CRISPR system into the cell we want to edit, along with an altered version of the gene. So the CRISPR system cuts the original version, and the cell repairs the damage by copying the altered version. So our idea was, well, how about we simply encode the CRISPR system right next to the altered gene, such that when the cell repairs the damage to the original gene, it will copy both the altered gene and the CRISPR system. That way, the offspring of that organism will inherit the altered gene and the CRISPR system, and they'll also inherit from the other parent a copy of the wild-type gene, the original version. CRISPR will then act again to cut the original gene and copy over the altered gene and the CRISPR system. This way, the CRISPR system effectively makes that same edit in every generation, even in, potentially, the wild.
And based on your own modelling studies, how quickly could that propagate through a population? Because that will be an absolutely critical thing if this is to be an effective control against malaria.
So it depends on how many altered organisms we release relative to the size of the original population, and also on how fast the organism reproduces. Insects like mosquitoes that breed very, very rapidly, we could potentially alter all of them in the span of only a few years.
What about the environmental consequences, negative impacts, and also the rebound of the strong selective pressure you're going to put on the pathogen? Will it not come back with a vengeance with some strategy to surmount your CRISPR system? It could evolve an anti-CRISPR gene, for example.
It could. Fortunately, we have 2,000 or so different variants of the CRISPR system that bacteria have provided us, so we could probably simply switch to a new one. But in many cases, what really prompted us to write these papers before we actually demonstrated this in the laboratory was because we realized that malaria was, while an excellent target, it's really only one potential application. And we know that CRISPR works in every species we've tested throughout all the kingdoms of life. And this is a very general strategy that can be applied to alter almost any sort of gene. So what this potentially means, these CRISPR gene drives, we could alter almost any sort of sexually reproducing populations in the wild. And that could have some pretty profound ecological effects. That's why we're going to have to use it very, very carefully. And we're going to have to be sure that we involve everyone who might possibly be affected. Because we all rely on healthy ecosystems, and we all share a responsibility to pass them on intact. So if we have an ability to alter them by changing entire populations, then we're going to have to be very certain that we do this as safely and responsibly as possible.
Kevin Esveld. That's it for this month. Don't forget that all of the papers we've been discussing are freely available alongside previous editions of this podcast from eLife's website. That's at elifesciences.org/elife. My name's Chris Smith. This is a Naked Scientists production. You can find out more about us from our website at nakedscientist.com, and I'll be back in September for another look inside eLife. Until then, thank you for listening, and goodbye.
The eLife podcast from eLife, the open access journal for outstanding research in the life and biomedical sciences. Online at elife.elifesciences.org.